So, ready for our next talk. Paulo Amorim from UFAL will tell us about the life of ants. Thank you for coming.
Thank you, Bojan. And thank you, of course, to the organizers for the kind invitation to be able to present my work here. So, I will talk about a continuous model of ant foraging with pheromones and trail formation. And a part of this work is in collaboration with Ricardo Alonso from PUC and Thierry Goudon from Nice. So, of course, I don't have to show to anybody what is a foraging trail, because, I mean, everybody who has once been a child knows what an ant foraging trail is. But to get pictures of ant foraging trails, it's nice to live in Rio, because I can just almost step outside of my apartment and see these huge trails without any vegetation. And of course, here there is no scale, but this is a trail like more than 20 or 30 centimeters wide, which I photographed just here in Aterro do Flamengo. And this is also taken by me of one of these little ants, not so little, even though it seems little, which was dead, of course, otherwise I wouldn't pick it up, because one of them bit me and it was really painful. So these ants in particular, they don't sting you, they actually bite with their huge jaws. And it can be quite painful. But this is just to say that I'm really happy to be working with studying ants and also living in Rio, because if sometimes I forget about my work, I am instantly reminded of my work every time I leave the house. And not only when I leave the house, but also inside my kitchen, where these little ants that maybe the people here who live in Brazil more generally, and especially in Rio, probably know. The very small, almost white ants known as ghost ants, which are the bane of kitchens everywhere in Rio. They just seem to appear out of nowhere. That's why they call them ghost ants, I think. But more seriously now, why do people study ants? Well, of course, besides scientific curiosity, many ants, like this one, the fire ant, Solenopsis invicta, and some other species, are considered invasive, dangerous, or pests in some parts of the world, in particular in the United States of America, where they have been introduced by human commerce and migration, and where they were not native, and they can displace other species and cause major damage to the ecosystems. So, of course, it's important to study ants because of these factors. So what about ant foraging? Well, ant foraging, which we all know from a direct observation of ants, is actually one of the most interesting phenomena in the animal kingdom, a collective emergent phenomenon. Because if you think about it, ants generally have no leader and nobody to organize their behavior, except, of course, for the queen. But the queen is not telling the ants to go search for food or do this or do that. She's just laying eggs. So it's a situation where we have individuals that follow simple and local behavior rules and which produce complex, rich, seemingly intelligent, large-scale strategies. This is just a close-up of these leaf-cutting ants that I showed you before, where you can see that they have these very large mandibles. And these are everywhere here, even here in Rio, in the more green areas. So, okay. Now, you may think that okay, maybe ants are smart because they can do all these organized behavior. And here is one ant which has a huge head, which is its huge brain. So it's a very intelligent ant. But actually, no, these are just its skull, its head only houses the large muscles that it needs to open and close the mandibles to cut through the vegetables. So they are not very smart, each one of them in general. So each one of them in particular have very weak vision and very limited cognitive ability. So how is it that they can originate complex and organized behavior at the collective level? Well, of course, this is a question that has been partially answered by many biologists along the ages, so I will just present my small modeling contribution to this type of questions. Okay, so of course, ants must communicate in order to produce complex behavior at the collective level. So how do they communicate? Actually, ants have been found to use nearly every means of communication available. Tactile communication, so they touch each other with their legs or with their antenna. Visual communication, so they can stand up more or less and assume different attitudes which represent different situations. Chemical communication, which is what we will be concentrating on, and even acoustic communication. This is not very well known, but sometimes when there is an underground nest and a portion of a tunnel collapses, they will, some species of ants can produce sounds that the other ones will detect to know where they are. But one of the most important means of communications which ants use are, of course, pheromones. Pheromones are chemicals that the ants produce and leave on the substrate. And they are used mostly for the communicating many things inside an ant colony and outside of it, like to signal alarm, the presence of food, or colony-specific olfactory signatures so that they can recognize nest mates. And even the queen uses pheromones to inhibit the worker's sexual development or to let it happen when the colony is producing alates, which are the fertile females and the males. So what about foraging dynamics in general? Well, a very naive view of foraging dynamics, but which really contains most of the information, is this. So of course, from the ant's point of view, the goal is to efficiently discover and remove food sources. The key point is that ants are attracted to foraging pheromones. Well, of course, many ants, not all of them, but we will concentrate on these. And so pheromone laying can be easily imagined to produce a useful feedback loop for the ants. For example, ants can find food through random foraging. When they find food, they return to the nest carrying food or after having consumed the food. And when they return to the nest, they deposit pheromone. The other ants, which are either lying around or in the nest, detect the pheromone and are attracted to it. So they follow the trail to the food source, and then they reach the food again and repeat the process. And the trail is reinforced in this way. If, by some chance, they follow the trail to the food source, but there is no longer any food, they will not return to the nest laying pheromone. And so the pheromone will just degrade or evaporate, and the trail will be quickly abandoned. So this simple mechanism has the potential to explain, in general terms, how ants forage. In a very simplified way, of course. Okay, so what we wish to do is find a mathematical model where this sort of simple rules can be translated into some system of partial differential equations which produce trail formation. So there are many possible approaches to this model, to this type of situation. The most natural one, of course, is to use discrete models where we simulate all the little ants and give them rules for their behavior, and this exists, and it is very interesting. But continuous models, like the one that I will present, exist only until very recently for isolated parts of the foraging dynamics and not for the whole cycle of foraging, let's say. So this is what I will try to do. So let me just present a little quite basic background material on modeling animal foraging, which has to do, of course, with the diffusion equation. So how can I deduce in a very elementary, very naive way some parabolic equations, let's say, or some partial differential equations which are simple, but which can model the movement of ants or other animals for that matter? Okay, so we suppose that the ant moves randomly on a one-dimensional territory. This is really background material, but I think it's not a common deduction of the diffusion equation, so let me just present it. And the ant starts from x equals 0, and I denote by p x of t the probability of finding the ant at time t at point x. And at each time increment, it moves to the left or to the right by the amount delta x. Then, of course, what is written here is the basic law is that of a kind of a Brownian motion in the line, is that if the ant is at position x at time t, then either it was at position x minus delta x before, or at the position x plus delta x. So either it came from the right or either it came from the left with equal probability. Of course, this is all formal, but just to see what is the equation. So we have this probability, then we do a Taylor expansion. This is very simple, plug into our main law, throw away the terms that we don't like, which will supposedly be small in the limit, and obtain what is, of course, the diffusion equation or the heat equation, okay? So in n dimensions, you just get the usual heat equation with the diffusion coefficient and the Laplacian. Okay, so this, of course, there are millions of more sophisticated ways to do this. This is just how you explain to someone who doesn't know that parabolic equations or the heat equation can also be modeled, can also model the movement of animals. Okay, this is more or less the very elementary reason why that may happen. So more generally, you can deduce using or motivate using the same type of arguments a more reaction diffusion type equation to model ants, to model animal movement in general or any other thing. So I will look at this p function as a density of whatever it is that I'm trying to model and not as a probability. And in general, we will work with equations, with reaction diffusion equations, which have this general form. So the rate of change is given by diffusion due to random movement, a transport in the direction of some vector field v. So if your transport term is of this form, you know that the ants or whatever it is you're modeling have a tendency to follow the given vector v, and then a source or sink term on the right-hand side. So ultimately, we will be interested in ants following pheromones. So this velocity v, I can think of it as being the concentration gradient of this pheromone, of some chemical. So this is what we will want to reach. And of course, it turns out that this very basic idea already exists in the scope of chemotaxis. So the chemotaxis system is let's say, maybe the simplest possible system which has this sort of phenomenon. It is used to model aggregations of bacteria, slime mold cells, and many other things. And in this canonic Calorcito, so-called Calorcito model, what we have is the following. So here in this notation, rho is the density of bacteria, and c, small c, is the density of the chemoattractant. And so this model contains a random motion of the bacteria, let's say, because of this Laplacian, and a transport term, which is telling me that the bacteria are attracted to growing concentrations, to increasing concentrations of the chemical C. The chemical C also solves a parabolic equation which says that it diffuses somehow through the substrate On the other hand, this term gives the evaporation rate or degradation rate, but this term gives me that the chemical is produced by the bacteria itself. So the bacteria create a chemical and they are attracted to that same chemical. That is the gist of the chemotaxis system. So of course this has been introduced, I don't know, maybe in the 70s or 50s, I don't remember, and it's been widely studied along with thousands of variants, but some conspicuous mathematical properties can be enumerated, like possible blow-up of solutions for this type of system, depending on spatial dimensions and the size of the initial data, which is, again, easy to understand why it might happen, because the bacteria produce a chemical, they are attracted to the chemical themselves, and of course this can originate a feedback loop which originates finite time blow-up. Okay. So what we propose finally is a model which is of chemotaxis type to model ant foraging. The key point is that our population will be represented not by individual ants, but by density functions of time and space, and will be divided into two groups. One is the foraging ants, denoted by U, and another one is the returning ants, denoted by W. The pheromone concentration here will have the letter V, and the food distribution will be C. So V is the pheromone. And what we will try to incorporate in the equations is the following simple dynamical rules, which mostly I had already said before. So the foraging ants emerge from the nest, then disperse randomly with a tendency to follow the direction of increasing pheromone if they find any pheromone. Once they reach the food, they transform into returning ants and collect the food. Returning ants return to the nest and deposit pheromone as they go along. When they reach the nest, the returning ants transform into foraging ants, and the pheromone diffuses and evaporates. Let me just point out some modeling assumptions that we make. So perhaps one of the most important ones is that we prescribe a homebound vector field, which we denote by this gradient. So this may seem a little bit artificial, but actually, in this context, there's probably no other way of doing it. Because, okay, so when the ant reaches the food, finds the food, then it has to go back to the nest. And what I assume is that it knows how to go back to the nest. It's information which is contained in the ant's brain. But this is actually what happens in the literature. What I mean is they don't use some sort of emergent phenomenon like following some pheromones or something to go back to the nest, typically. No, they use orientation by visual landmarks, the CO2 concentration around the nest. I mean, people do these experiments where you have the nest and the ant is returning and there's this rock by the nest, and they take the rock, they put it somewhere else, and the ant goes to another place. So they really use like traditional means to orient themselves towards the nest. What I mean is it's not an emergent phenomenon. So this is prescribed. And the dynamics near the nest and the food are very, very simplified in this model. So the nest is just a special area of the domain where the returning ants transform instantaneously into foraging ants with a certain rate. So there's, in reality, there's a lot of stuff going on inside and around the nest which we simply do not model. And the food side, also the same thing. It's just a special area where we say that there is food and where the foraging ants transform into returning ants and deplete the food. Okay, and one experimental fact which we also take into account and which is actually important for us is that pheromone deposition decreases as ants approach the nest, at least for some species. So this we take into account. So finally, here are the equations in a more complicated form, which I will simplify in a moment. Let me just point out that we have 4 equations, one for the foraging ants, another one for the returning ants, another for the pheromone, and a final an ODE for the food concentration, food distribution. Okay, so let's look at the non-dimensional system, which appears a little bit simpler. So again, just looking at one of the equations, we have the foraging ants move by random dispersal and follow increasing pheromone, because we see here the gradient of V, and V is the equation for the pheromone. And on the right-hand side, we have the interchange between the foraging ants U and the returning ants W. So one of them disappears at the food and appear as foraging ants. And at the nest, the same thing. This capital N is a given function which tells me where the nest is located. And you can ignore this last function. It just describes emergence from the nest. Okay, so the second equation is the same, but the returning ants follow the given homebound vector field. The pheromone is just a normal diffusion equation where we see that it is produced by the returning ants with this spatially varying rate P of x and the food removal. We impose no flux boundary conditions just to conserve the total mass of ants, which is an important property, of course, which should be valid with our assumptions. So let me present some numerical simulations of this model. We use an upwind finite difference scheme for the spatial derivatives and a fourth order RK scheme for the time evolution. And we can see the main phenomenon that we're trying to get, the trail formation to and from the nest. So here is the results for one simulation of foraging ants, where they start out, so this is 1, 2, 3, 4. They start out getting out of the nest. And then they are here following these trails to food, which is not indicated here, but which is at the end of these sort of trails. Okay. So with more contrast, this looks better. Then for the returning ants, we can see them as the foraging ants start discovering the food. They start to appear near the food, and then they form a trail towards the nest. Here there are 2 food sources, so here one is being depleted and the other one has just been discovered, and in this frame the first one has already been depleted. Okay, so again, the fact that the returning ants form a trail towards the nest is written into my equations. It's not surprising, it's nothing. But this, what I call trail formation by the foraging ants, is only indirectly implied by the equation, so this is what we observe. Pheromone concentration more or less accompanies the formation of the trails, but with a little bit more dispersion. And here is the food being eaten by the ants. Here is the the evolution of the food quantity, and we see that it's depleted in about 5.5 hours, which with the data that I fed into the model for these simulations is more or less realistic. Just an example with some topography, which makes it look a little bit more realistic, but the idea is the same, and a fancy graph with some colors. Okay, but it's the same idea, also with topography. Random topography. Okay, so how much time do I have? Okay, thanks. So let me just present a very quick, quite basic parameter exploration to find conditions in the parameters of the equations which favor the formation of trails numerically. So what we did was, in this big mosaic, in the horizontal axis you have the evaporation rate of the pheromone, and the vertical axis is the chemical sensitivity of the foraging ants. Okay. And okay, so we can see the result of various simulations. And these white and black stars, which concentrate along this sort of diagonal here, indicate for each row or for each column the minimum value of time taken to eat all the food. So from the point of view of the ants, where there is a star among this parameter space is where they will be able to get all the food more quickly, and so evolutionarily it will be better for them. And what we observe is that all those points are concentrated along a sort of line, and they coincide with the places where, at least looking at the picture, of course, trail formation is more apparent. So this somehow suggests, through a very simple parameter exploration, that indeed, which is not surprising, trail formation is advantageous for the ants, and also gives some possible relations between these two parameters. We could have chosen another pair, but to concentrate on these two, a sort of relationship between the two parameters which favors trail laying. Okay. And what's important is that these parameters are controllable by the ants. I mean, through evolution, of course, not by the individual ants. Sensitivity and the evaporation rate can be changed by natural evolution, by natural selection. And so, of course, this is just a very simple exploration, but it somehow shows that we can, theoretically at least, say something concrete about what values of the parameters we ought to find in a particular species of ants. So, okay, so let me just quickly present some analytical results. This part is in collaboration with Ricardo and Thierry Goudon. First, let me talk about what we call the fast pheromone diffusion foraging system, which is a slight simplification of the system that I presented. So here we only have three equations where we suppose that the pheromone diffusion rate is very fast relative to the ant's movement. And this gives that the equation for the pheromone, which now is called P, is just an elliptic equation. So the pheromone is produced by the returning ants W, and the foraging ants U are attracted to the pheromone. And here, sorry for the change in notation, but here the V is now the given homebound, nest-bound vector field. There is no food source anymore except for this given function C of x, because I suppose that the food is either very large or renewable, so there is no depletion of food in this time scale. N of x is the given nest function, and as I said, delta V is the direction to the nest, which is also a given. Okay, so one of the main results that we obtain is existence and uniqueness, a well-posedness result for weak solutions for this system, which in particular proves these formation of integrability results and formation, let's say, of boundedness. This is also so-called a hypercontractivity result. So what this says is that even when the initial data is not in an L-gamma space, there is creation of the L-gamma norm for positive times with this precise rate. And the same is true for L infinity. So even if the initial data are not bounded, they will instantaneously be bounded according to this rate. If the initial data are bounded in these LP spaces, then we just obtain a usual boundedness estimate on these LP spaces. Okay, so of course the main one of the main steps in proving something like this is to prove some a priori estimates. So let me just talk a little bit about this. How can we prove an estimate of this sort? And before that, let me just point out that according to these results, there is no blow-up in finite time. However, in the usual chemotaxis system, which I write here also in its parabolic elliptic form, there may be blow-up in finite time in 2 dimensions, which is the case we are interested in. So why is that? Well, very formally, this may happen in the usual chemotaxis system, because if you do some calculations, and formally, you get for the density this sort of equation. So if you ignore these spatial terms in this one, which will not make the solution explode, for sure, if we ignore the spatial terms which just make the solution move around, we have this sort of Riccati ODE with the time derivative equals to the square, which of course is a function which can, or an equation which originates blow up in finite time. So this very formally is the reason why we can expect blow up in systems of chemotaxis. In our system, however, if you do the same type of very formal computations, what you find is that the derivative in time of u plus some spatial terms, some spatial variation terms, grows according to not u squared, which would be the same situation as before, but u times w, where w is the density of returning ends. But w itself is the solution of a parabolic equation with a drift term which is given. So it's natural to expect it to be somehow regularized, and so that u times w is better than u squared for our estimates. So this is indeed what happens. So how do we prove the a priori estimates? Well, we use as usual tools like the GNS inequality in 2 dimensions in this form. The mass conservation is crucial in all of this, which is a consequence of the structure of our system, and some classical Lebesgue interpolation results. So the main step maybe is establishing this type of inequality, which is just a consequence of multiplying the equations by the appropriate powers of the functions and playing around with all the terms. And we see that we have some different powers on both sides. So just to present a very simple analysis formally where in which the domain is bounded so that we can do it much more easily, we just use Young's inequality of this type. Take care to take the 2 parameters of the 2 exponents inside a certain range. And okay, so we get this, and we can see that we can absorb the appropriate, so the blue terms will get absorbed on the left-hand side, and the magenta terms will get absorbed on this left-hand side once we sum the 2 inequalities. And so we get an inequality like this. And here we recognize that we can use a sort of ODE comparison result for this quantity after we have taken care to use some Lebesgue interpolation to, let me just go back, to turn this term into a term with these powers, but with a higher power on the outside. So this is just to do a rough sketch what we do here. So transform that last estimate into a sort of ODE inequality, and use a comparison result to compare the solution with this sort of power. So, and then you have to work a little bit with the exponents, but this is essentially the desired result. So this is more or less how the a priori estimates work. It's just more sophisticated Gronval, actually. Okay, so for the L infinity estimate, there may be various ways of doing it. Upon putting some restrictions on the nest function and on the food function, it's possible to do a simpler Moser iteration. But for the general case, the Georgi energy method for level sets also works. So let me just sketch very rapidly how it works. We define, for W is our unknown, and we define these level set functions. And this sequence of levels and times is a bit technical. And of course, the energy functional for the level sets. So this L2 type energy. And the work is into proving a recurrence relation for the WK, which gives that they converge to 0. And since this type of integral is smaller than UK, we conclude, after some work to prove this sort of recurrence relation, that actually the function vanishes above a certain well-chosen value m, and so sort of uniform bound is obtained. So this is an adaptation of the Georgi level set method. For the complete fully parabolic model, so almost exactly the model that I presented in the numerical simulations, we also have results in this paper which is submitted, but using totally different techniques, namely working directly with the heat kernel. So, thank you for your attention.
Thank you, Paulo.